So the topic was biomedical signals. Okay. So last time we discussed about various biomedical signals. I will just take a fast recap and then we'll go to the next point, that is action potential. So last time we have discussed different types of biomedical signals which are available from body. Now today we will discuss about what do we mean by action potential. This is one of the important concept. How exactly the electrical potential it is generated in our body. So what is the phenomena? What is happening behind? So that all we'll discuss today. Okay. So this all we discussed last time. Different types of biomedical signal. Now when I say biomedical signal, so this includes all different types of signals. Okay. Now, in our body, there are bioelectric signals, there are bioacoustic signals. That means the all different types of sounds our body is making while its normal working is going on. So those are bioacoustic signals, like the valves of the heart, right? Then next it is biomechanical signal. So our different types of organs inside our human body, so what mechanical actions are taking place, so those signals can be extracted and those are biomechanical signals. Okay. Then biochemical signals. Now in this case, different chemical reactions keep on happening in our different organs of the body, and the chemical measurements can be taken from that, and that those gives us some information, useful information for diagnosis or analysis purpose, and those type of signals are biochemical signals. Okay. Then biomagnetic signals. So last time we have discussed some of our organs produce very weak magnetic field. Okay, so those type of signals are biomagnetic signals. Then biooptic signals. So there are some optical signals produced by our biological systems, like our human eye. So those are biooptic signals. Then bioimpedance signals. The impedance of the tissues, it changes as per the different conditions, like the composition, that means the water content, blood distribution, and the volume, all those things. So it decides the impedance of these tissues, and measurement of impedance that gives us valuable information for analysis as well as diagnosis purpose for the researchers as well as for the doctors. So those are bioimpedance signals. Then this diagram we have discussed last time, which gives us the list of all the different types of biomedical signals. Okay. Then here in this diagram we have discussed about what are the corresponding frequencies and what are the corresponding amplitudes of all these different types of bioelectric signals. These also we have discussed. Okay. Then sources of biomedical. Okay. Now this list we have discussed last time, that actually what are biomedical signals we have discussed till now. So bioelectric signal, it is one of that, right? There are mechanical signals, there are optic signals, there are magnetic signals, and so on. But as as our subject is concerned, for us the for our subject, the important signal is bioelectric signal. Okay. So these are all different types of bioelectric signals: ECG, EEG, EMG, ERG, electroretinography, electrooculography, and so on. So these are the frequency ranges and the type of transducers required. Right? That means the electrodes required for this. So these electrodes we'll discuss in more detail in the next lecture. Okay. So this is all just introduction of what are different types of biomedical signals and bioelectric signals. So this table gives us the different types of bioelectric signals. Now the question is how actually electricity is generated in our body. Okay, so that is what we are going to discuss today in details. Okay, So our today's topic is how the electricity is generated in our body, okay, which gives rise to different types of bioelectric signals, or called as bioelectric potential. So what are the sources of bioelectric potentials that we are going to discuss in detail, and what are the corresponding phenomena? Okay. Now the bioelectric potentials are produced because of some electrochemical activity. So our body, there are different cells, different organs in our body are made up of different tissues. Different tissues are made up of different types of cells. Okay, and all these different types of cells, they have different chemical compositions. Okay, and some chemical activity, it is always going on in our human body cells. Okay, so as a result of the chemical activity, some electric potential is generated, and that we call as the electrochemical activity. Okay, so this electrochemical activity, it gives rise to some potential, and this happens because of some cells called as excitable cells. Okay, now all cells in our body are not, do not produce any potential. Dead cells, they do not produce any potential. Okay, so the type of cells which give rise to this bioelectric potentials are some of the cells in our body, which we call them as excitable cells. So which can be excited, and because of this excitation, some voltage changes, potential electric potential changes happen inside the body, and that's why those are called as bioelectric potential. So that we are going to discuss. Okay, now the type of excitation cells in our body are related to the nervous tissue, related to the muscular tissue, and the glandular tissue. The different types of glands which which are responsible for secretion of the various types of hormones or different chemicals for different activities in our body. So those are glands. Okay, so the cells related to nervous tissues, muscular tissues, and glandular tissues, these are the excitation cells, and these cells usually produce some electric potential. Okay, now to go in further detail, if you consider a neuron, if you consider the nervous system, right, there is axon, dendrites, and neuron. These right, these are the different parts of this neuron in our nervous system. You might have seen introduction of this in the neural network subject. Okay, so if we consider a very small part of this, which is shown here as in the expanded version. Okay, now if this is considered as a single cell, then in the resting state, this cell, it has a negative charge inside and the positive charge outside of the body, outside of this cell. Okay, now if we insert some electrode, what is shown here, if we insert some electrode inside the cell, and if we connect some voltmeter connecting the outside of the cell and the inside of the cell with the help of some micro electrode, then some potential is found to exist here, right? Now during resting state, this potential is around minus 70 volt. This minus 70 volt, it is the typical value. It cannot be exactly minus 70 volt for each and every human being. So it is some typical value. Usually this may be in the range of around 60 to 100 volts, not volts, millivolts. Okay, so what I have written here, this is the typical value. So with it is possible to measure the voltage with the help of some microelectrodes. Micro means the thickness is very small, which can be easily inserted in our cell. Okay, so this potential we call it as the resting potential, which is under, or you can call it as the potential under steady state. Resting potential. We'll discuss about all these things now in further detail. Okay. So electric field is generated in cells, for example, nerves or muscles and organs because of intra and extracellular ionic currents. So this electric field is generated because there are some ionic currents are flowing inside this cells. So that's why it gives rise to some electric field. And the ionic currents, these flow because of some electrochemical process. So the electrochemical process, this gives rise to some ionic currents, and these ionic currents finally they produce some electric field. Okay. So some cellular structure which is shown here, this I am going to discuss in more details now. Now what you see here, the blue dots, these are different types of ions, maybe positive ions, negative ions, and these openings what you see here, these are the channels, or these are also called as gates. These are the channels or the gates which open or close for certain conditions. These are the gates or channels. Now we'll discuss now how these channels open and close and how actually the currents flow and so on. Okay. So this is how the electric signal is generated. Okay. Now as I discussed in the previous slide, there is a minus 70 millivolt voltage between the inner side of the cell and the outer side of the cell. Okay, so that is called as the resting potential. Now this potential is produced, we have seen, only in case of excitable cells or the cells which is having excitable membranes. Membrane means it is the outer covering of the cell. Okay. Now some energy is expended in expelling sodium ions and potassium, drawing potassium ions. So as I told earlier, there are some positive particles and the negative particles, the charged particles, so called as the sodium ions and the potassium ions, which keep on changing their position, and accordingly the potential inside the cell keeps on varying. And this for the movement of the sodium ions and potassium ions, some energy is expended. Okay, so that is what happens. Now because of this, it gives rise to some electric and concentration gradients. So as this, because of the movement of the sodium ions and the potassium ions, which are the charged particles, it gives rise to electric concentration and concentration gradients across the cell membranes, and which in turn produces some field. Now when the cell is stimulated, that means it is excited, the permeability of the cell membrane undergoes the cycle of changes. So this means the cell membranes, so cell membrane means it is the outer covering of the cell. It is the outer covering of the cell. So this is the cell membrane. Now the permeability of the cell membrane keeps on changing as per the stimulation. Now, because of the change in permeability, the ions, they move inside or they come outside of this cell membrane, and because of that some ionic currents starts flowing. Okay, so this happens when the cell is excited. Okay, so when the cell is excited, some permeability of the cell membrane undergoes changes, and one cycle of electric potential it takes place. Now we'll see what exactly is this cycle, how it is produced, and so on, that we are going to discuss today. Okay, and this the occurring of this cycle of the potential, it is all or nothing phenomena. It is all or nothing phenomena. That means the complete cycle will happen or nothing happens. Okay, so if some trigger is given to this cell, complete cycle of the potential it will be produced, otherwise nothing is produced. So that's why it is called as all or nothing phenomena. That means the cycle triggers in full or not at all. There cannot be half cycle like that. Okay, so this cycle of changes can also trigger other responses from the cell on top of the polarization cycle. Okay, now when we consider this cycle, there are actually two parts: the resting state and the action state. So we'll see what do you mean by this resting state and the action state. So resting state means when some excitation is given to the cell. Now this this excitation, it may be electrical excitation or it may be mechanical excitation. Okay, and because of that some action takes place, and during this action the potential the electric potential it will change. Okay, so there are two states of the excitation cells: resting and action potential. Okay, so this is similar to the earlier diagram what we have seen earlier. So this is the inner part of the cell and this is the outside part of the cell. Now inside part of the cell there is an intracellular fluid here, and outside there is an extracellular fluid. Okay, and this is a cell membrane. This is the cell membrane. That means it is the boundary between the inner part of the cell and outer part of the cell. Okay, and along this boundary you will find there are different types of channels. Okay, and the properties of these channels keep on changing as per the excitation. So when this channel opens, then in that case there will be movement of the charged particles either from inner to outer side or from out to inside. Okay, so this cell membrane, what we are talking about, this is actually having a semi-permeable characteristics. So it is made up of semi-permeable lipid bilayer made of lipids and proteins. So this is the semi-permeable layer made up of lipids and proteins. These are actually some biological names. Lipid means these are some this material, it is soluble in alcohol and it is not soluble in water. Soluble in alcohol and not soluble in water. So that material is lipid. So this cell membrane, it is made up of lipids and proteins. So that separates the inner part and the outer part of this cellular structure. And the permeability characteristics keep on changing as per the excitation. Okay, so the cell membrane is a very thin with a thickness of 7 to 15 nanometer. So the thickness of this cell membrane, it is in the range of 7 to 15 nanometers. Okay. Now the transmembrane ion channels allow flow of ions across the membrane. So these channels, what we have discussed earlier, so these channels allow a flow of ions from the membrane, through the membrane, either from in to out or out to in. The cell membrane is a thin dielectric material which acts as a charge separator. So as we have seen, there is negative charge inside, there is a negative charge inside, and there is positive charge outside of this cell. This happens during the resting potential, resting state. Okay, so this cell membrane, it acts as charge separator. On one side the charge is different, on the other side the charge is different. With a dielectric constant of epsilon equal to 5 and the typical value of capacitance, it is 0.5 to 1 microfarad per centimeter square. The cell membrane is impermeable to intracellular protein and other organic anions. So it does not allow flow of intracellular protein and other organic anions. So anions means it is the group of atoms that have gained electrons, which have additional electrons. Okay. Now the cell membrane, as we have seen, it is impermeable to intracellular protein and organic anions. That means a group of atoms or radicals. Okay. Whereas it is semi-permeable for sodium, potassium, and chlorine ions. So this is very important. The cell membrane, that means the partition between in and out of the cell, it is semi-permeable for sodium, potassium, and chlorine ions. And because of this, the flow of current takes place actually. So that is what we are going to discuss. Okay, so the separation of charge due to selective permeability of the membrane ions is responsible for membrane potentials. Okay, so this sodium, potassium, and chlorine ions, these are responsible for membrane potential or for some ionic currents. Okay, so that is what we are going to discuss. Now, the movement of the sodium, potassium, and chlorine ion, it happens because of the concentration difference across the membrane, which creates a diffusion gradient. And because of the diffusion gradient, these ions, they move from one side to the other side, giving rise to some current as well as potential. So movement of ions, which results in electric currents traveling along the membrane. And the ion flow by diffusion create a potential difference, okay, which is similar to p-n junction here, which is similar to p-n junction. Now the current in electric circuits is the flow of free electrons, whereas in case of this biological phenomena, the electric current, it is the current flowing here, it is because of flow of free ions. So these free ions are the ions which we have just now seen, the chlorine, sodium, and potassium, positively charged ions and negatively charged ions. So the movement of these ions in the semi-permeable membrane, it gives rise to flow of current. Okay, so these are all basic things related to our cellular structure, the ions and so on. Okay, now we'll see what different things we are going to study related to this. I already told you there is some potential called as the resting potential. Okay, so and that value also I told you it is around minus 70 millivolt. When the cell is not excited, it is under resting state. So that is called as the resting potential. And when the cell is excited, just now we have seen, it gives rise to some cycle of potential, and that is called as the action potential. So this action potential now we'll discuss. Okay. Then now during this change from resting potential to action potential, some depolarization and the repolarization happens. So we'll see what exactly you mean by depolarization and repolarization. Okay, now as I told you, there are different types of ions: the sodium ions, potential ions, and the chlorine ions. Okay, sodium, potassium, and chlorine is Cl. These two are positively charged ions, and the chlorine is negatively charged ion. Now, inside our cell membrane, there are some channel gates. I told you. So there is a sodium channel. Okay, so potassium channel gate, and there is some sodium channel called as the sodial pump. So some pumping action happens, and because of this pumping action, there is a movement of sodium and potassium ions. So that is called as the sodium pump. Okay, and this also I told you, it is a all or nothing law. That means the complete after trigger, either the complete cycle takes place or nothing happens. So that's why it is called as all or nothing law. Okay, then we'll see when this action potential is generated, what is the peak to peak value of this action potential. So that is called as net height of the action potential. Then what do you mean by absolute refractory period? So when you say refractory period, that means it is along the time axis. So for example, on x-axis, if there is a time here, it is a time here, and y-axis, it may be a voltage, induced voltage developed because of the excitation. Okay. So this absolute refractory period, this is along the time axis, whereas this net potential that will define, it is along the voltage axis. Similarly, relative refractory period along the time axis. Then propagation rate. So these three things, these are along the time axis. Propagation rate, per Nor conduction rate, or conduction velocity. Okay, so these are various terminologies we are going to discuss in this presentation. Okay, now again, let us come back to the membrane. Now membrane is which surrounds the neuron. Okay, so as we have seen earlier, it is composed of lipid and protein material. So this is the membrane. This is the membrane, and this yellow color things, these are some channels or the gates. These are channels which open and close, giving rise to movement of sodium and sodium potassium ions. Okay, now these channels, we'll see how they work, right? So this is the membrane, and these are the channels. So membrane, as we have seen, it is made up of lipid material and the protein. Lipid, I told you, it is a material which is soluble in alcohol but not soluble in water. Okay, then I also told you this is the membrane, and the membrane which separates the inner and outer side of the cell. Okay, so this is the inside and this is outside of the cell. Now during resting state, inside it is negatively charged and outside it is positively charged. So outside it is positively charged, and if we measure this, now usually the measurement we always take uh, measurement of the inside potential with reference to outside. So that's why we denote it as minus 70 millivolt. Okay, so it is the electrical charge across the membrane called as the membrane potential. So the resting potential is around 70 millivolt. Difference between the inside and the outside, it is minus 70 millivolt if it is measured inside with reference to the outside. Okay. Now, as I told you, inner side it is negatively charged and outside it is positively charged. So positively charged, these are some sodium ions here which are positively charged, which are more in number outside, that's why it is positively charged. And these chlorine ions, these are negatively charged ions. Sodium are positively charged. And okay, now similarly inside this cell there are potassium ions and organic anions. So anions are the group of ions having which have gained electrons. So that's why they are negatively charged. So overall there is more negative charge inside and as compared to outside. That's why the inner potential we say it is minus 70 millivolt. Okay, so ions are electrically charged particles, sodium, potassium and chlorine ions. Okay, so the resting potential exists because ions are concentrated on different sides of the membrane. Sodium and chlorine are outside the cell, and inside the cell there are potassium and organic anions. These exist inside the cell. That's why there is negative potential here and positive potential outside. Okay. Now under steady state, when there is no excitation, sodium ions are actively transported to maintain the resting potential. Okay, so this is some channel through which the transfer of sodium and potassium takes place. So under resting state, these sodium ions, these are expelled outside. Okay, so that's why on inside overall charge it is positive and outside overall charge it is positive. Inside it overall charge is negative. Okay, and there is a sodium potassium pump which exchanges 3 sodium ions for 2 potassium ions. So if 3 sodium ions are moving out, you can see here if 3 sodium ions are moving out, 2 potential ions they move in. So that is how this sodium potassium pump works. That is the working of this sodium potassium pump. Okay. Now opening of ion channels. Now these channels, as I told you, these channels they keep on they either open and close as when the cell is excited. So the opening of ion channels leads to depolarization, okay, and which makes an action potential more likely by some excitation. So when some excitation is given, because of the opening of the ion channels, depolarization happens. That means the sodium it will be expelled outside. So that's why, okay, after opening this, the sodium it will move in. So that's why the negative charge inside that will decrease, and this is called as depolarization. Inside of the postsynaptic cell becomes less negative because of the movement of sodium ions inside. Okay. Then opening of the ion channels leads to hyperpolarization, that means overpolarization, which makes action potential less likely, and this is called as inhibitory postsynaptic potential. Inhibitory means which is trying to avoid this excitation. Okay, so inside of the postsynaptic cell becomes more negative, and okay, because of the movement of potassium and chlorines. Now, in general, the integration of both things happen. That means the exhibitory and inhibitory PSPs. The integration takes place, and that is the overall potential. Okay. So inhibitory PSPs will counteract the effect of exhibitory PSPs at the same neuron. Now, the summation of both these phenomena, it gives rise to the overall potential. So this potential, if there is sufficient depolarization at the axon hillock, an action potential will be generated. That means if exhibitory potential it becomes more than inhibitory potential, then in that case it gives rise to depolarization, triggering the cycle of action potential. And we call it as the firing of the neuron. Now the action potential is the rapid depolarization of the membrane. Now exactly what is this depolarization, polarization, and all those things, I will explain you with one diagram which is going to come. Okay. Now during this process of depolarization, see, normally before depolarization, inside it is negative, outside it is positive, right? Now when some excitation takes place, then the sodium ions which are outside, this channel opens, and this charge, these sodium ions, they start moving inside. Okay, and that is what we call it as the polarization, depolarization. Okay, now let us see what is this. Now when partial depolarization, okay, see, normally negative, inside of the cell it is negative, outside of the cell it is positive. Now when the cell is excited, then in that case the sodium, these sodium channels, this is the channel which opens, and these sodium ions, they move inside, and because of that the inside of the channel it becomes positive, because the positive charge now it will be more. So that's why inside becomes positive and outside becomes negative. So we this we call it as depolarization. So this is what happens after excitation of the cell, because these sodium channels, these are voltage gated sodium channels. These are voltage gated sodium channels. Okay, so sodium ions rush inside, and because of that the potential here, which was minus 70 millivolt, potential here which was minus 70 millivolt, because of the rush of these sodium ions inside, this changes to plus 40 millivolt. So this becomes plus 40 millivolt. So this is called as depolarization. Okay, so this phenomena, it is called as a depolarization. So the sodium, positively charged sodium ions, these are moving in, and because of that the voltage here, which was minus 70 millivolt, it becomes more and more positive. So this is depolarization. Okay, Now, when this depolarization takes place, as the sodium ions they move inside, depolarization takes place, and this reaches to some saturation point. Okay, so it was minus 70 millivolt, right? So it becomes more and more positive. At some point, it will saturate, and after that, it starts again becoming more negative. So that is called as repolarization. So again, inside of the cell, it becomes negative, and outside of the cell, it becomes positive. So this is called as repolarization. Okay. So sodium ion channels close. That means these channels they close. So after these channels are closed, then again it becomes negative inside and positive outside. That means resting state. Okay. So the depolarization triggers opening of voltage gated potassium ions, potassium channels. So this is actually a potassium channel. So as inside becomes positive because of the flow of the sodium ions, so this positive charge inside, this opens the voltage gated potassium channel. So this is a voltage gated potassium channel. When the voltage becomes positive, this channel opens and the potassium it is forced to go outside. So because of that, again this becomes negative and outside becomes positive. So the potassium ions rush out of the cell, repolarizing and then hyperpolarizing the membrane. Okay. So this is what is repolarization. So this part, this is repolarization. Okay. So this is the complete cycle of this action potential. So action potential, as we have seen, it is all or none. That means the complete cycle takes place or nothing happens. And it is always of the same size. Either it is triggered or not at all. 2-liter depolarization or the membrane is refractory. Okay. So it is triggered. If it is triggered completely. So these are some characteristics of this. Now this complete cycle, it is indicated in this diagram. Okay. So A, this is part A here. The action potential begins with the partial depolarization. So there is a partial depolarization here, and because of that, the action potential begins. Now, as the depolarization starts, it becomes more and more positive. Okay, and that part, it is this B. Excitation threshold is reached. There is a sudden large depolarization. Okay, and after that, it reaches to certain peak, and when it reaches to certain peak, the sodium voltage control gates they open, and the sodium is expelled outside, and that is called as repolarization. So this is repolarization. Okay, now after it is repolarization, suddenly it will go below minus 70 millivolt, and that is called as hyperpolarization. So that part it is called as hyperpolarization because there is excess negative, right? Then after certain time, it again goes to the normal state, that means the resting state, which is minus 70 millivolt. Okay, so that is refractory period, which follows immediately after the action potential, where no depolarization can occur. So this is the complete cycle of this action potential. And this same phenomena happens for the nerve cell or the muscle cells or in general, any cells inside the body which generate electricity. Okay. And this action potential, it keeps on transmitting from one cell to the other cell, and that's why it gives rise to some electric current, as you can see in this diagram. Okay, so this is the diagram from our book, which is mentioned in our syllabus. So resting potential, depolarization, action potential, repolarization. Okay, and after potentials. Okay, so I think we'll stop now. So next time we'll again discuss a little about this action potential, and then we will move to the next topic. Okay, before I stop, any questions, any queries? If you have, I will take some questions. Otherwise, we'll end this. Those who have joined late, give your attendance in the chat box, and all are requested to join at the right time. Please avoid joining in the middle of the lecture. Okay, everybody has given attendance, and any questions? Any query anybody would like to ask? Do you find it interesting, biomedical? Because all these things actually we see, because right till now most of the subjects we are studying just theory. Now we are coming to more and more application oriented subjects, which you can feel, we can carry out some measurements and those things. Okay, but all these actually we'll see very elementary things. These are very big fields actually, and if you find interesting later on in your post graduation or PhD, whatever you do, you can explore these subjects in more details.